quick look for IB biology. We're now moving on to look at chains of amino acids called polypeptides and their structural organization. Our first consideration is going to be what is called the primary structure, and then we'll consider that against the other types of structure which we'll be studying in later videos. There's a PDF in the description for you to annotate your own notes, and there's a quiz there as well for you to check your understanding of the content of what we're doing here. The primary structure of a protein is the order of the amino acids in the polypeptide chain. In the example shown in front of you, I have drawn a 1, 2, 3, and 4 amino acid chain, and the amino acids in this chain are all glycine. So this is my primary structure. 4 amino acids, all of them glycine, and this is called the primary structure. In later videos, we'll see how the R groups interact and how these polypeptides fold up into secondary structures, which is the next video, and then we have another video on tertiary and quaternary, which is more complex folding of proteins. So let's look at this primary structure in just a little bit more detail. So this is the same structure that I showed previously, 4 amino acids, each of them glycine, and this would be abbreviated to GLY, 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 and GLY as a way of recording the primary structure. Notice that there's an amino group at one end and a carboxylic acid group at the other one, known as the N-terminal and the C-terminal. And that each of the amino acids is joined to each other by a peptide bond, shown in red here. And that the backbone of the molecule follows the sequence nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. Amino acids can join together in chains from 50s or even way up into the thousands. As examples, there's insulin, which actually has 51 amino acids joined together in a chain, whereas titin, which is found in muscle structure, has something like 26, just short of 27,000 amino acids in a particular order. So the order is very precise. The order of the amino acids in a polypeptide is actually dictated by the genetic code. So we have this complication and this potential for tremendous diversity, that we have 20 different amino acids, and we can organize and arrange them and join them together into chains of tremendous length, short and long, and we can change the sequence of those amino acids in the chain. So this potential is huge. And in fact, the total number of different amino acids that a person have is called the proteome. So these are all the different proteins, different types of protein that you have, and currently the estimate for human beings is round about 100,000 different proteins operating within our cell chemistry. Now there's something of a paradox here, because the order of the amino acids is dictated by the genetic code, and yet the genetic code has current estimates at 20,000 genes. So of course this is our genome. So how can it be that 20,000 genes can code for 100,000 proteins? And of course, those are the details that will be found in our study of the nucleic acids and in protein synthesis. The primary structure is the order of the amino acids in a polypeptide. The links below are leading us to looking at secondary structures, subscription buttons there for you, and also a link to the channel playlist on proteins.